So welcome to the Arthropod Genomics X, the AGSx, our session number 3, featuring the Beenome100 and Comparative Genomics, B-Genomics that is. We've run this several times in the past. I think this is our 3rd or 4th year. I'm kind of starting to lose track already. So we have a little bit of information to kind of go up with you over here at the front of the end of the seminar. And a little bit of information as far as the QR code in the very top hand right that you can access for finding additional information about the present speakers today. So just to let you know, this is a joint effort between the USDA ARS, National Institute of Health, NIH, and the I5K, the international consortium kind of aimed at sequencing 5,000 arthropod genomes. We are through sessions 1 and 2, and thank you for everybody who attended those. We're up to session 3 now, featuring Beenome100 and Comparative Genomics. This is brought to you and organized by Jay Evans and Michael Branstetter, both from USDA ARS. And our session number 4 will be coming up in May on the 21st. So definitely check out the link in the QR code to sign up for that as well. Going over the Comparative Genome Resources, NCBI's new toolkit for data and tools for unlocking arthropod genomics research. And that's brought to us by Terrence Murphy. And this is made possible by many people, not just those in front of the camera today, but everybody who kind of working in the background. And so definitely want to give a great big thanks to our national program leader, Kevin Hackett, for condoning this and making this all possible. Myself as the lead organizer, Glenn Haynes, the one working in the background for Zoom, getting the registration forms and kind of making everything run very smoothly in totality. Anna Childers, who does the website and puts out all the Twitter feeds for us, as well as handles recordings that get uploaded into YouTube. Forgot that on there. Pia Olsen, who has been running all the recordings of our presenters very, very smoothly. And Rob Waterhouse, who runs the Slack channel. Some additional resources kind of mentioned just on the previous slide. There's an informational website, has the link as well as that QR code once again. There is the registration form for if you have not signed up for session number 4, please go ahead and do so right there. Is also accessible through the webpage. Our YouTube channel has the link right there. And I'll be dropping these all into the chat for you during the session, so you can kind of click on those and don't have to copy and paste anything over. As well as the Slack channel. That does require a registration, so we can send out a separate link for you to join that as well. So at this point in time, I'm going to turn it over to Jay Evans, who's going to be kind of leading the session from this point onwards. Thanks, Jay.
Great, thank you, Brad. Yeah, we're really excited about the speakers today and this series. They, again, will be recorded if you or your lab mates wish to catch up in the future. But it's been a great series. Last year's and the prior years on these topics have also been recorded. Some of those speakers are actually online now as well and might help with the questions. But we do need to get going. These are pre-recorded talks, but the speakers will be in this virtual space to help address any questions. Please put those questions in the Q&A rather than the chat, and we will monitor that and try to fit them in. It's going to be a very tight schedule, so it's you know, if we don't get to them here, we'll address them by email afterwards. So hopefully you've registered, in which case your emails will be on file and you'll get a roundup and links to the recording. So without further ado, let's start. The first speaker will be Dr. Michael Branstetter, again from the USDA ARS Pollinating Insects Research Unit in Logan, Utah. And his talk is Beenome100, Bee Diversity Through a Genomic Lens.
Greetings and welcome to the first talk of the Arthropod Genomics Virtual Symposium. My name is Michael Branstetter and I am a research entomologist with the Pollinating Insects Research Unit in Logan, Utah. It is my pleasure to introduce the series by providing an overview of the Beenome100 project. Before I begin, I would like to acknowledge Ligia Benavides, Rena Schweizer, and Catherine Paris. They've contributed material to this talk and helped with some of the analyses, so I want to make sure that they're acknowledged. This talk represents accumulated knowledge and analyses from several years of Beenome effort, with many people sort of contributing to this, as I'll mention later. So the goal in this talk is to provide an overview of the Beenome100 project, starting with some justification for why bees and why bee genomics, moving on to a summary of the origins, objectives, and general procedures of the project, and ending with some results, starting with a general progress update, and then presenting some updated genomes for bees of agricultural importance, as well as a first look at bee genome size evolution across the tree of life. So why focus on bees as a target for genome sequencing? I think first and foremost, it must be said that bees are extremely beneficial to humans as pollinators of flowering plants in agricultural and natural ecosystems, with bees providing essential pollination services to a wide diversity of fruit, nut, vegetable, and seed crops. Although much agricultural pollination is achieved by a small set of managed species, native wild bees are also vital. It is not an overstatement to say that bees are vital to human food security and that studying and managing bees is just as important as studying and managing pest insects. But why sequence more than one bee genome? Is it enough to have sequenced the honeybee genome? No. Bees, like many insect groups, are very diverse. They're represented by over 20,000 species globally and at least 4,000 in North America, excluding Mexico. Bees vary greatly in size, shape, and behavior, and they exhibit a wide range of life history traits, including kleptoparasitism, host plant specialization, and sociality. As we'll hear about later in the session, bees are an exceptionally important group for understanding the evolution of sociality, given that the trait has evolved repeatedly in different lineages. Like many insects, bees are under threat from multiple stressors, and many species are in decline, with 8 species currently listed as endangered and many more being considered. So conservation is also an important consideration for why we want to study bees and bee genomics. Developing more complete genomic resources for bees is valuable for a variety of reasons. One of the most important is for the health of bees, a topic reviewed in a recent paper by Grotzinger and Zayed. In this paper, they discuss the value of genomics for understanding bee immunity and detoxification, and for help with bee breeding and strain identification. Genomics also facilitates phylogenomic and evolutionary research and is useful for conservation genomics. Having genomes of diverse bees could help us predict the sensitivity of species to pesticides, breed more robust and efficient pollinators, and provide genetic resources that help us evaluate and conserve bee diversity. So how are we doing in terms of generating reference genomes for bees? Well, until recently, not very good. As of 2020, there were only 18 published bee genomes representing a very taxonomically biased set of genomes, with most coming from social lineages in the families Apidae and Halictidae. Given that the honeybee genome was first published in 2006, why is the accumulation of bee genomes rather slow? The answer is that there are many challenges to getting a good reference genome. High cost, limited access to fresh material, small specimen sizes, and lack of computational power have made generating reference genomes a challenge. However, these challenges are starting to go away as the sequencing costs decrease and the molecular and computational tools improve. The graph here shows the accumulation of bee genomes and NCBI over time. The first bee genome was published in 2006, the honeybee genome, and between 2006 and 2020, a few new bee genomes have been released. But as of 2020, the rate of new genomes has exploded, likely thanks to the new technologies for sequencing, as well as large-scale, efforts, consortium, such as the Earth Biogenome Project, the Darwin Tree of Life, and now the Beenome100 Project. So what is the mission of Beenome100? As stated on the project website, the mission of Beenome100 is to sequence, assemble, and annotate high-quality reference genomes for 100 plus USB pollinators, while also strengthening resources for the honeybee, a key pollinator and insect model. Using the latest sequencing tech, a major goal of Beenome is to expand the taxonomic coverage of bee reference genomes and to improve the quality of existing genomes for basic and applied research purposes. The Beenome100 project didn't just arise out of nowhere, but it's sort of building upon past work led by USDA and others. So key projects include the ongoing AGPest 100 initiative, which aimed to sequence 100 plus insect pest genomes. This project was led by Kevin Hackett, Anna Childers, and Scott Geib, and others at USDA. And AgPest 100 provides, it's really the basis for Beenome100. So a lot of the the sort of workflows and infrastructure for Beenome100 are really benefiting from what AGPest100 has developed. But Beenome also adds to old and new genome sequencing projects like I5K and the Earth Biogenome Initiative, which aims to sequence all species on Earth. So Beenome100 is led by Gene Robinson at the University of Illinois, as well as Kevin Hackett and Jay Evans of USDA. The project started when Gene helped acquire congressionally mandated funds to conduct honeybee and native bee genomics research. USDA was brought in to help lead the project and disperse funds to various USDA labs and collaborators that could help collect and identify native bees. And the project now includes over 14 USDA labs and university collaborators. Beenome100 is organized into groups with project leads and participants. Groups include bee diversity and conservation, data generation, processing and management, genome structure, sociality, and detoxification. The general idea is to generate the genomes and then have each group work on topic-specific analyses, culminating in a series of linked papers and projects that will hopefully be of high impact. Later in today's session, you'll hear from leaders in the conservation, sociality, and honeybee groups. One of the key challenges for the project has been how to prioritize and acquire material for sequencing. I have personally been involved in the diversity or taxon selection group and have helped determine what species to target for sequencing. A major consideration has been to acquire a taxonomically diverse set of bees at the tribal and generic ranks, but we've also tried to consider other features, including agricultural importance, conservation status, geographic coverage, life history diversity, and sociality. Feasibility has also been a major consideration due to the challenges of getting fresh material. So properly collecting, identifying, and vouchering bees for genomics is challenging due to the fact that specimens need to be flash frozen in order to preserve high molecular weight DNA. And so our general procedure has been to net bees in the field, take voucher images while specimens are still alive, freeze specimens in liquid nitrogen, and then ship off specimens for sequencing. We also tried to collect a series of specimens so that at least one individual from the series can be maintained as a voucher specimen that gets properly deposited in a major research collection for future review. Unlike other organismal groups, it is important for bees to carefully check the sex of specimens, given that females are diploid and males are haploid. We have generally tried to target haploid male specimens for sequencing, but we generally take whatever we can get. Once specimens are collected and frozen, the Beenome team selects species for submission and usually sends them to Hilo, Hawaii for processing in the USDA ARS Pacific Basin Agricultural Research Lab, where Scott Geib is the research leader. Some of the sequencing is also being carried out in Stoneville, Mississippi by Brian Scheffler and his genomic lab, but most of the non-apis bees are going to Hilo. This slide presents the general sequencing and data processing pipeline that we are using for Beenome. Many people have contributed to this workflow, but I would like to especially acknowledge the contributions made by Scott Geib, Shenna Sim, and Anna Childers. Scott's lab initially specialized in extraction, DNA QC, and library preparation, but after acquiring PacBio and Illumina-like sequencing instruments, his lab has been doing the sequencing as well, which has improved project efficiency greatly. To generate gold standard reference quality genomes, we aim to sequence all species using 3 different approaches. PacBio Hi-Fi is being done for the main genome assembly. The recent development of low and ultra-low library preparation kits has enabled us to use small amounts of tissue. In most cases, only single specimens are needed, or even parts of specimens, for adequate Hi-Fi sequencing. For scaffolding and genome curation, we are generating Illumina Hi-C sequence data. And finally, for annotation, we are doing RNA-seq, preferably using multiple libraries from different tissues. However, what can be done in terms of these different kinds of sequencing depends on the bee and the number of specimens available. Due to limits on tissue availability, some species only get sequenced for Hi-Fi or Hi-Fi plus Hi-C, but even Hi-Fi only assemblies tend to be much better than genomes of the past. The data processing workflow is iterative and loops around depending on results and availability of Hi-Fi, Hi-C, and RNA data, as well as the sex of the bee specimens submitted. As a quick overview, we assemble the Hi-Fi data using Hi-Fi ASM, We scaffold the assembly using the Hi-C data and the program Hi-C-Yaz. We extract mitogenomes using mito-hi-fi, and we annotate genomes using NCBI's annotation pipeline, combined with new or existing evidence, such as RNA-seq data. We prefer to make genomes public and use NCBI's pipeline so the annotations are consistent across genomes. We're also using a variety of tools to QC the raw data assemblies and curated genomes to catch and fix problems along the way. One of the most important tools is blob tools, which allows us to tag and remove microbial contaminants from the genome assemblies. These data are of interest to the microbial group, who will be analyzing the microbial bycatch across bee species. So now switching to sort of progress and results. So far, we have submitted, or sort of collected and submitted, over 176 bee species for sequencing. Taxonomically, we have covered all US bee families, all tribes, and over 70 out of the 112 US bee genera. On the data processing front, we have some data for 143 of these species that we've collected. And of these, 66 genome projects are considered done and either uploaded to NCBI or are ready to be uploaded. 2 genomes have been published as notes so far, and several more are close to being submitted. As you can see, Beenome100 is well on the way to generating 100 plus reference genomes, and we anticipate having 100 new genomes on NCBI in the next, probably 3 to 6 months. The slide here provided by Catherine Paris shows the geographic coverage of sampling for Beenome. Most samples are from the US, but a few bees have been sent to us from Canada and also from Panama, where there are some interesting social bees that will be of use to the sociality subgroup. The figure here, generated back in November of last year, provides a phylogenetic perspective of our coverage. So this tree, based on ultra-conserved element data, incorporates existing bee genome data on NCBI, that's shown with black text, and then new bee genomes generated by Beenome, and that's in the blue text. So bee families are color-coded. So looking at the branches, the different colors represent the 7 different bee families. And a major takeaway from this figure is that we have greatly expanded the taxonomic coverage for bees, based on our sampling just from the US. In addition to our sort of standalone website, Beenome100 is now featured on the GOAT website, or Genomes on a Tree. This is a really great tool which provides detailed information about what species Beenome has targeted, as well as the progress of those genomes. And so we just got this set up in the last month, and we'll be updating and maintaining it monthly. So you can come and track the Beenome progress here. Over the next few slides, I will present some of our exciting new results, starting with improved genomes for bees of agricultural importance. In addition to the honeybee and bumblebees, several solitary bees are used in managed pollination. 3 of these are the alkali bee, Nomia malanderi, the alfalfa leaf-cutting bee, Megachile rotundata, and the blue orchard bee, Osmia lignaria. All of these bees have had their genomes sequenced previously using short-read instruments. Here I present our updated genomes for these species and compare our results with the earlier ones. So shown here in the first slide is the genome of the alkali bee, Nomia malanderi. This bee is an important pollinator of alfalfa in the western United States. The original genome, published in 2019 by Kapheim et al., produced a scaffold-level assembly that was 326 megabases in length and had an N50, which is a measure of sort of genome quality, of 2.4 megabases. Our new Hi-Fi plus Hi-C genome has produced a chromosome-level assembly that is 449 megabases in size and with a scaffold N50 of 18 megabases, significantly improving the contiguity of this important pollinator. We also have evidence from Hi-C data that there are 14 chromosomes. And shown on all of these sort of species, these genome vignettes, are a picture of the bee, the stats that I just talked about, as well as a snail plot from the program Blob Tools, the Hi-C curation map in the middle, and a blob tools plot on the right, showing sort of a combination of target and non-target DNA. Next is Megachile rotundata. This is also an important pollinator of alfalfa in the US and other places. There was an unpublished genome generated in 2011 using short read sequence data. And this genome has a size of 272.2 megabases and an N50 of 1.7 megabases. Our new Hi5 plus Hi-C genome is chromosome level with 16 chromosomes, and it has an N50 of 5.96 megabases, demonstrating that this is a larger and more, this new genome is larger and more contiguous than the previous one. Finally, Osmia lignaria, the blue orchard bee, is a solitary bee that is a managed pollinator in almond and fruit orchards, and is often used as sort of an alternative pollinator to the honeybee. An unpublished genome was generated several years ago using Illumina and actually some PacBio data, and this genome has a size of 177 megabases and an N50 of 5.5 megabases. Our now chromosome level assembly has 16 chromosomes, and it is 499 megabases in length and has an N50 of 14.9 megabases. So as you can see, all of these three genomes using the newer technologies have a significantly larger and more contiguous genome, as well as we have sort of added information about the number of chromosomes in the genomes from the Hi-C assembly information. So here I'd like to highlight an interesting issue that we've observed in our data. So this figure shows the Hi-C scaffolding maps generated using Hi-C YAS and visualized with JuiceBox. Many of our maps look like the one on the right, in which the core genome, shown in red, makes up half or less of the total assembly size. Whether or not the non-mapping repetitive regions are removed from the genome has a big impact on the total final genome size. So for Osmia lignaria, the genome size is either 217 megabases if you remove those repetitive regions, the non-red parts, or 499 megabases if you leave it in. And so one of the things we've kind of struggled with initially was, do we leave this in or remove it? And it seems like in some cases people do remove this repetitive DNA. And so one of the things we did is started to look to see, if we can get other information about the sort of structure and size of the genomes in some bees. And so we found some old karyotypes for species of Osmia. And in these, it has been shown that Osmia species tend to have a lot of heterochromatin or repetitive elements in their genomes, suggesting that the large amount of repetitive DNA we are observing is real and not an artifact of sequencing or sequencing and assembly or contamination. The karyotype information also confirms that Osmia species tend to have 16 chromosomes, which is the number we recovered from the Hi-C data. This is also shown by comparing the genomes of several other Osmia species. So the genomes of four different species of Osmia, Osmia lignaria, Taurus bruneri, and Osmia bicornis, all have 16 chromosomes based on the Hi-C mapping data. And they also have a similar amount of repetitive DNA in the assemblies, although these figures show the repetitive regions mostly removed from the assemblies. So we're getting some good sort of verification of our results between different genomes as well as the karyotype information. So that's it for the agricultural bee genomes, and kind of just showing you sort of the quality of the genomes and how they've improved from previous sequencing. But so the last kind of part of my talk is sort of looking at the evolution of genome size across bees. And so one of the subgroups is kind of looking at genome structure in bees. And I think a sort of initial, kind of part of that is just looking at genome size. So here we compiled data on genome size from NCBI and from our new Beenome data. And this figure summarizes bee genome sizes across the phylogeny of bees and with the data organized by species as well as by family. So what we see is that the mean bee genome size is 402 megabases with a range from 177 megabases on the smaller end to up to 1.8 gigabases. Interestingly, many orchid bees and a few other bee lineages have, significantly enlarged genomes above, 1 gigabase, but the average bee genome seems to be below 1 gigabase, as far as we know so far. For the most part, bee genome size seems to be consistent across families, although the validity shown in orange in the bottom right graph potentially have significantly larger genomes. The only issue is we don't have very many mylidid genomes sequenced yet. Using the same data set, one thing we wanted to ask is that do genomes of parasitic bee species have significantly smaller genome sizes than non-parasitic bees? This question was asked in a recent genome note by Sless et al. In 2022, which was describing a single new parasitic bee genome. And this general idea has been sort of discussed in the literature, the idea that sort of parasitic species have reduced genomes compared to non-parasites. And so we looked at this by comparing, or by sort of compiling all the data we have, and we actually have genomes for 16 parasitic bees now across multiple lineages, and we compared these to the non-parasites. And what we find is that although parasitic bee genomes tend to have a smaller genome size, the result currently is not significant, with the mean parasite bee genome size being 307 megabases, and the mean non-parasite being 410 megabases in size. However, it is kind of cool when you look at sort of the chromosome structure across these parasites. Although it's not found in all of them, we do see that many of the parasitic bee genomes appear to have reduced numbers of chromosomes, as shown in these high-C contact maps. So here are sort of 3 different parasitic bees from 2 different families. And in each case, they have 6 chromosomes. And so as you might recall we don't really have a good sense of the sort of mean chromosome size in bees yet, but on average, bees seem to have, more than 10, similar to the osmia, around 16. And so fewer than 10 is on the smaller size in terms of the number of chromosomes. So that's pretty much it for my talk, but I think, stay tuned for much more to come. We're just sort of wrapping up phase 1 of Beenome in the next few months, and hopefully moving on to phase 2 in the future, which will be to add, more diversity. And so before I end my talk, I'd really like to thank all of the people who have contributed specimens to Beenome. They're pictured here. And I definitely want to acknowledge, all the people that come to our monthly meetings and contribute, ideas and time and discussion. We're a very open and collaborative group, and anybody is sort of welcome to come and join or to collect and submit material for future sequencing. And so I would definitely like to acknowledge the sort of Beenome leadership of Jean, Kevin, and Jay. I would like to acknowledge Scott, Shenna, Brian, and Anna for the help with all the sequencing and data processing, and then all of the various subgroup leads and other people that participate in those groups. Thank you so much, and a quick shout out to Nina Sokolov, who has provided us with an awesome potential logo for our Beenome project. We still haven't decided on an official logo, but we have some great artwork as a possibility for the project. So thank you so much. Please check out our websites or email me.
Okay, I think that was the end of the video. Michael is online. I don't see any questions in the Q&A, but certainly address them to him. But I think in the meantime, there's my areas in person. If there are any pressing questions, please drop them right now. Otherwise, we should probably move on to the next speaker. If you could pull up Rena's talk. Great. Okay, well, let's do this one, and then hopefully have time for general questions for the project at large. So Dr. Rena Schweizer is going to speak about the Beenome100 Project and enabling bee conservation in the omics era.
Hello, my name is Rena Schweizer and I'm a research entomologist postdoc with the USDA Agricultural Research Service Pollinating Insects Research Unit. I'm really grateful for the opportunity to be here today to present some of our ongoing research as part of the Beenome100 Project on developing tools and conducting research for the conservation of bees. Wild bees are critical to the health and productivity of the world's natural and agricultural systems. Yet many bees are declining due to disturbances, pathogens, pesticides, habitat loss, and climate change. Many of these stressors can have especially large impacts in small, isolated populations that don't have the genetic diversity, population numbers, or gene flow to counteract the effects. Understanding and preventing the biological aspects of the decline of bees takes an integrative approach. And this means that we assess all aspects of a bee's phenotype, from the DNA level to the whole organism level. And we can use a multitude of omics type data approaches to examine and measure each level of this hierarchy. The interactions of all of these biological aspects, in context with the environment, can help scientists quantify how a bee responds to climate stressors, pathogen load, and other environmental factors. My talk today will focus on the utility of one particular omics data type to help us understand and conserve bee diversity, and that, of course, is genomics. Genomic data provides insights into key population parameters that are critical for assessing and managing species of conservation concern. For example, genomic data provide insight into parameters such as identifying inbreeding, gene flow, and understanding demographic history. Genomic data can also be used to understand bee fitness, or the genetic underpinnings of decline, resilience, and health. Genomics is critical for management and breeding, such as identifying management units, aiding selection for resilience or for traits of interest, and performing genetic rescue. And finally, genomic data sets provide reproducible, efficient, long-term data for monitoring and assessing species recovery. And many, but not all, of these genomic approaches require some sort of reference genome. However, a minuscule percentage of all bee species have publicly available genomes on NCBI. So this plot shows the number of genomes by release year. You can see that there's an upward trend in the number of genomes, in part due to improvements in technology and global genome sequencing initiatives. However, keep in mind that there are around 20,000 bee species worldwide, with around 4,000 in the US alone. So this is a really small fraction of the world's bee diversity. The Beenome100 initiative was formed in part to help fill this gap in reference genomes for bee species. The initiative was formed in 2021 as a coordination across several USDA ARS research units and university collaborators. The mission is to sequence, assemble, and annotate high-quality reference genomes for 100 or more US bee pollinators, while also strengthening resources for the honeybee, which is a key pollinator and insect model. And of course, in picking the species to be included as part of Beenome100, there's been a focus on native bees of conservation, management, and agricultural importance, as well as representing a broad diversity of bee species. Our approach for Beenome100 has been de novo genome assembly. So with other ARS research units, we are generating high molecular weight libraries and transcriptome data. We're using long-read sequence data from PacBio, as well as high-C data for chromosome structure. The transcriptome data is being used for annotation of genes in these genomes. We then assemble the genomes using scalable, reproducible workflows, and then submit the genomes to NCBI for annotation and public release. And of course, we're developing conservation genomics projects using these new reference genomes, of course with many other non-conservation goals as well. We currently have around 170 species in various stages of sequencing and assembly, so we're on our way to exceeding Beenome100. Taxonomically, we've covered all US bee families and over half of the US genera. This figure shows a phylogeny of bees generated using ultra-conserved elements. In blue text are around 90 genomes that we have in a finalized assembly state, and the black are genomes that have been uploaded to NCBI. Several of the species for which we've generated reference genomes under the Beenome100 project are species of concern, and here are some photos of these very charismatic mini fauna. I've highlighted with a star if they are on the IUCN Red List, or a triangle if they're listed or being considered for listing as endangered under the U.S. Fish and Wildlife Service. There are several bombus species, of course, as well as other species from across the bee phylogeny. Of course, when undertaking this immense effort to sequence genomes, we have to ask how a reference genome itself could aid conservation-related goals. So we can learn about the genome structure, the gene content, and other characteristics for a particular species, and this tells us about bee fitness. We can use the reference genome for resequencing studies, which again can give us those essential conservation parameters as well as be used for management and breeding. We can use the reference genome for identifying sequences of important markers, genome-wide or mitochondrial or locus-specific for environmental DNA monitoring. And then we can use the associated sequence data from a reference genome to identify the microbiome or associated organisms, which again tell us something about the bee fitness. So today I'll briefly tell you about two Beenome100 conservation-related projects. The first will be how we've generated a novel de novo reference genome for a species of conservation concern, which is the Mojave poppy bee. And then I'll tell you about how we're using a reference genome of a related species to aid the conservation and learn more about Franklin's bumblebee. So the Mojave poppy bee, or Perdita meconis, is a species of conservation concern that's restricted to the eastern Mojave Desert of North America. It's a specialist pollinator of two poppy genera and is being considered for listing under the U.S. Endangered Species Act, along with one of its pollinator hosts, the Las Vegas bear poppy, shown here in this beautiful photo. So you can see it's a really small bee. Adults are anywhere from 4 to 7 millimeters. It has a really short active period, and it's an extreme specialist on these poppies. So these characteristics make it a really special, cool bee, but also can make it a little bit hard to find especially during these really short active periods. So much of the survey work for Perdita meconis has been led by members of the USDA Bee Lab, including Terry Griswold, Michael Branstetter, Colleen Might, and Utah State faculty Joe Wilson, among others. And a few years ago, it was suggested that a reference genome for the Mojave poppy bee could be useful for subsequently characterizing population genetic data that could then be used for species status assessment. Using DNA from a single male specimen, the Beenome100 team generated long-read sequencing data, and we subsequently assembled a high-quality, near-chromosome-level de novo genome. The quality of this genome is fairly remarkable given the really small size of the organism and the fact that at the time it wasn't properly preserved in the field in ideal conditions for long-read sequencing. The genome is around 330 megabases. You can see on the right in the snail plot figure details of the N50 and N90 length for the genome, as well as the fact that we have around 95% of Hymenoptera genes complete. We compared aspects of the Mojave poppy genome to those of other published bee genomes. You can see the genera of them written at the top of the slide. The genome size and GC content fall within the values of those of published genomes. The number of protein coding genes, however, is at the high end for published genomes. This annotation is based on orthologous sequences only, though, and we're working on collecting additional specimens for RNA-seq data to verify the annotation and hopefully get a better idea of whether or not we're capturing the true number of protein coding genes or whether or not it's a technical artifact. What's exciting for the world of bee comparative genomics and for the conservation of this species is that the genome represents one of only a few genomes in Andrenidae, which is a highly diverse group of bees. And the Perdita genus is the most diverse genus in North America with around 640 species. So this figure shows results from a phylogenetic analysis, again using ultra-conserved elements extracted from published genome sequences on NCBI, as well as our new Mojave poppy genome, shown here with the star. And so this genome really does represent a newly sequenced, unique evolutionary branch of the bee phylogeny. This reference genome is now a resource for planned conservation work. Thanks to recent field collection efforts by members of the Bee Lab, we have a second specimen that was used to collect Hi-C data that will be used to scaffold the genome to chromosome level. This reference genome is also being used by graduate student Colleen Might, who's planning to use low-coverage whole genome resequencing of bees from across the range to characterize population structure, assess levels of genetic diversity, and measure gene flow, basically to identify all those essential conservation parameters. Furthermore, we have a collaboration with the USGS to develop environmental DNA assays to detect this rare species on blooming bear poppy plants. So the photo on the right shows Michael swabbing the petals of a blooming poppy to try to collect eDNA samples. And we're also assembling a mitochondrial genome to help with designing molecular markers for these efforts. And of course, the overall goals are to not only learn more about this unique species, but to provide genetic data for the U.S. Fish and Wildlife Service listing decision. Next, I'd like to tell you a little bit about our work on Franklin's bumblebee, a highly endangered bee species, and how we're using a high-quality reference genome of a related species to aid conservation and identifying key population parameters of this endangered species. As part of the Beenome100 project, we've focused especially on some of the North American bumblebee species, many of which are endangered or threatened, as you saw earlier in my talk. A couple years ago, USDA research entomologist Dr. John Kocher led the release of a chromosome-level, high-quality reference genome for Bombus affinis, or the rusty-patched bumblebee. This genome is not only exciting for enabling the genomics work for the rusty-patched bumblebee, but it can also enable reference-based sequencing and genotyping for a related Bombus species for which we don't yet have a reference genome, or it might be more difficult to generate one. So one such species is Franklin's bumblebee, which is a putatively extinct species that is U.S. Fish and Wildlife Service endangered and on the IUCN Red List. The species has an extremely narrow distribution in southern Oregon and northern California in an area that's about 70 miles wide and 190 miles long. The decline of Franklin's bumblebee was first noted by the late Robin Thorpe, pictured here, who was a professor at UC Davis. He dedicated significant efforts to surveying for franklini and other pollinators and helped push the listing of the first bumblebee species, the rusty-patched bumblebee, or Bombus affinis, under the Endangered Species Act. Franklin's bumblebee has not been observed in the wild since 2006. And as I mentioned at the beginning of my talk, Bombus franklini is one of several bumblebee species in decline. So previous work from Cameron and colleagues has shown dramatic declines in some of the bumblebee species in North America, declines of up to 96%. Additional work by this group demonstrated that the decline of some Bombus populations have been associated with high infection levels of a pathogen, Nosemi bombi, and with concurrently low genetic diversity. So it's possible that the small population size of Bombus franklini and the small geographic range could have kind of predisposed Bombus franklini to decline Although in these previous studies, they were not able to obtain genetic samples to really explore the population genetics. And bumblebees may be more susceptible to inbreeding or low genetic diversity due to haplodiploidy and eusociality, leading to low effective population sizes. So we aimed to get at some of this lack of genetic information on Bombus franklini through these three questions. So first, we wanted to know whether or not we can generate high-quality genome sequences from museum specimens. Second, do Bombus franklini individuals exemplify genetic characteristics of small populations, which could have predisposed them to decline? And third, did populations of Bombus franklini have reduced genetic diversity due to their decline? And our hypothesis was that Bombus franklini likely declined due to a combination of small population size dynamics and potentially high pathogen infection rates. So we undertook a museomics, that's another kind of omics, for Bombus franklini. We performed whole genome sequencing from 31 museum specimens collected between 1950 and 1998. So this map shows the sampling location, the year, and the approximate number of individuals sampled from Oregon and California. And of course, these were specimens that were really kindly donated by the UC Davis Bohart collection. We targeted females because they are the diploid sex. So we sampled mid-legs from females, performed DNA extraction, library prep, and whole genome sequencing, and then processed sequence data following the GATK best practices workflow. And here we made use of that high-quality reference genome for Bombus affinis, which is the sister species of Bombus franklini, and use it as a reference for mapping. And these data are very much in progress. So while we want to do many analyses, I'm going to show you some of them only right now on population structure and genetic diversity. So we had aimed to get at least 20x coverage for each genome, which would have set us up for obtaining high quality hard-called genotypes. And we actually exceeded our goals. We got unbelievable sequencing coverage, obtaining a mean of 40x coverage. We had a moderate number of PCR duplicates due to additional PCR cycles during library prep, since these were museum samples. And then we did observe a correlation between the collection year and the number of reads. So we knew that filtering would be important for removing any potential conflation of data quality and collection year. From the raw sequence data, we performed stringent filtering in order to come up with a set of high quality, high confidence, single nucleotide polymorphisms, or SNPs, from across the genome. One of the first things we did was assess population structure using principal components analysis of 31 individuals genotyped at around 42,000 linkage prune SNPs. Shown on the left are the results of our principal components analysis. Individuals are represented by points, and points are color-coded according to their collection year or their sampling locality. If you're unfamiliar with principal components analysis, the general idea is that points that are closer to one another on this plot are more genetically similar. Interestingly, we see no clustering associated with geography or collection year, and this lack of clustering suggests weak population structure and low gene flow. And of course, we're continuing to examine the exact partitioning of samples in this PC1 and PC2 space. We also found very low genome-wide heterozygosity. So here on the left is a violin plot of individual heterozygosity values for our filtered data set of 31 individuals. Here we're looking at around 210 million sites. So the mean heterozygosity is 0.000377, and this is a really low value, and actually one of the lowest published values for many species. So on the right is a histogram of genome-wide heterozygosity values from 85 species collated in Johnson et al. 2016. The Bombus franklini is shown in the red star. and with other species highlighted by letters. So, island foxes and African cheetahs are kind of well known for having really low genomic diversity, and we see that the Bombus franklini are close to those values. We also measured pairwise kinship, which is the probability that two randomly sampled alleles from two individuals are identical by descent. So the heat map on the left shows that most of the individuals are related at the second degree, even when looking at within collection years. Our mean kinship is 0.139, which is actually greater than the value recommended by experts for avoiding inbreeding depression. And then finally, to get at our third question, we examined the relationship between genome-wide heterozygosity and collection year. So we had expected to see a decline in heterozygosity associated with the decline of population numbers. However, we found no significant decline. So our results showed that we can obtain high-quality genomes from 25 to 73-year-old insect specimens, which I think is really cool and really opens up the field of museomics and insects for conservation genomics. We found that yes, Bombus franklini individuals seem to have low genetic diversity and moderate relatedness, which is suggestive of inbreeding and low gene flow. And then interestingly, at least up to the point of decline, we do not see a significant reduction in heterozygosity. So it's actually possible that Bombus franklini may have already had low genetic diversity, which then kind of increased the susceptibility to disease and other threats. We have some really exciting analyses that we're working on right now. We actually have PacBio data for Bombus franklini specimen. It's a museum specimen, so the reads aren't that long, but we're going to try to do a de novo genome assembly. And then we're going to do analyses targeted at understanding the demographic history, as well as assessing runs of homozygosity, because these give a kind of timing to when the species may have lost genomic diversity. And then, of course, other things about selection analyses and related to the environment's effect on the genome. So I think it's rare to have insight into a small isolated population of a bee, and Beenome100 has really enabled these analyses. So our hope is that Beenome100 will continue to help bee conservation moving forward. So a related ongoing project that I want to mention, but I unfortunately don't really have time to share the details. We have whole genome sequence data from 66 Bombus affinis individuals from multiple conservation units as defined in the species recovery plan. And you can see them identified with black dots on this map. So our plan with these data is to characterize population structure, assess inbreeding, using a genome-wide approach, and then to identify genetic markers that could be used for long-term monitoring of this species as we, strive for the recovery of this species. And of course, this work is a collaboration with a really large team of Bombus affinis researchers. With that, I would like to acknowledge the large collaborative team of folks who have been involved in Beenome100 and the associated conservation projects, as well as the financial and computing resources provided by the U.S. Department of Agriculture. And finally, thank you again to the organizers of this AGSx symposium for the invitation to present today. And with that, I'd be happy to take any questions. Thanks.
Thanks so much. Thanks, Rena. Yeah, I think there's time for some questions, and Michael is online as well. I did note there were a few addressed in the Q&A that hopefully you've seen the responses to from Rena and Michael. Any others? I know the best way, you could use the chat, of course, but the best way would be to use the Q&A function. Rena, there was one, I don't know if you addressed it on the economics of all these museum specimens. That might be interesting for folks. Do you want to embellish your response there? Maybe it's more, I think you addressed the extraction cost, but you know, how feasible would it be, and how far back do you think people could go to get viable genomes from something in a box?
I think Michael could probably comment more on the total cost of the sequencing. We did the actual sequencing runs in-house at the USDA ARS in Hilo, using a relatively new sequencing technology that's a less expensive alternative to Illumina, which up to this point has kind of been the standard for short read sequencing data. And you know, just from my own experience, I think that how far back you can extract DNA from a museum specimen, such as an insect that was never preserved really with DNA extraction in mind, I think there's a lot of variation in terms of, you know, how exactly the specimen was preserved, whether or not there are any sort of chemicals or inhibitors that have gotten onto the specimen in the, you know, 80 years it's been in a collection and handled. So it seems, at least for museum specimens and insects, there's a lot of variation, but within other taxa, there's been, you know, success getting DNA from skin or hair, or, you know, drilling into a tooth of a skull in a museum.
Maybe just one quick thing to add, because I think it's people kind of ask that question broadly, like, how much does it cost? And it really depends a little bit on, you know, your target coverage as well as the genome size. And so if we were doing orchid bees with gigabase genomes, it would cost a lot more, you know, if we wanted 20x coverage. But for the bumblebees, which tend to have smaller genomes, or maybe average size bee genomes that are around 300 megabases, it's cheaper. So yeah, I think it was like $50 per specimen just for the sequencing, plus, you know, $10 to $20 more for the you know, library prep reagents.
Right. There were a couple more questions / comments. Rob Waterhouse has on the fly vetted the assemblies of 104 Apidae for their Buscometrics. This is the predominant measure of assembly and gene prediction quality for all genomes, but insects as well. And says they fared pretty well. 95% passed a standard metric, or sorry, the 78 passed the Buscometric of 95% completeness of genes being present in this ortholog set across insects. So that's a good sign. Arrow asked when the recordings would be available. Pia, do you want to quickly address that, or Brad, or where will we post these in the end?
Oh, yes, I can. They will be posted as soon as the editing is completed, which takes maybe about a week, maybe a week and a half or two. So they'll be up as quickly as we can get them, and we'll send out an email to everybody to inform them of when they're available.
Yeah, and that's another reminder. I think some of we sent out a direct Zoom link. If you did not register for the webinars, you might not get that email. So please get in touch if you want. Make sure you get those links. Ooh, it might actually be one that I could partly address from David. Okay, which is microbial sequence in your assemblies. As hinted by Michael, there are a lot of actually scaffolded sets of DNA that are not B, or at least not assembling well into the B. Some are repeats that were problematic. Yeah, we have a group keen on that that's open to new group members, trying to see if we can harvest any microbes from these genome assemblies and the RNA-seq affiliates to these species. So far, with 11 or 12 vetted it's hit or miss. There are some with fully assembled endoparasites, all effectively assembled. There are many with other DNA features. We've just looked at the DNA data so far, and it looks promising, but not consistent, I'd say. Like, I don't think we're getting a holobiont for these, partly because of collection strategies, but it was there will be some resources of that sort, and we certainly want to share those with whoever. Oh, great. Oops. Ooh, a quick note from Michael Killowald. I'll let one of you historical DNA folks answer that really quickly, and we'll move on to the next
Um, you want me to reply to that one live?
Oh, actually, sorry, Michael, could you do it, just type in and let's, I think, yeah, if you could pull up the next speaker, we should probably stick on schedule. I just realized we have 3 more great talks to go, and everyone will be online to address them. But the next speakers actually will be a pair of presentations on social evolution with the title of Harnessing Natural Variation to Study the Evolution of Social Behavior. And the first speaker will be Dr. Karen Kapheim from Utah State University.
Hi, my name is Karen Kapheim. I'm in the biology department at Utah State University, where my group and I are very interested in using genomes to understand how social behavior evolves in bees. And today I'll be telling you about a relatively new area of research on this topic, where we're trying to understand the link between diapause and social evolution. And so before I get started, I just want to acknowledge my co-author and collaborator in this project, Dr. Priscilla Santos, who's a postdoc in the lab. She and I have developed a lot of these ideas together, and she ran most of the analyses that I'm going to show you today. So let's just get all on the same page about what diapause is. It's a period of suspended development in an insect, especially during unfavorable environmental conditions. And so the figure on the left is showing you what is typical for a solitary bee, where we have a new female emerging in the spring. She lays eggs, they hatch and develop as larvae until that last instar, the 5th instar larvae, also known as a pre-pupae. That's when they enter diapause, or this suspended development, and they spend most of the unfavorable period in that way, whether in the temperate region, that may be a winter or the hottest part of the summer. In the tropical regions, it may be a rainy or a dry season. Once the appropriate cues tell them to, they will break diapause and complete metamorphosis to emerge and start the cycle over again. So what does this have to do with sociality? Well, it's long been recognized that there's a link between diapause and eusociality. This has mostly been formalized by Jim Hunt almost 20 years ago, looking at the role of diapause in caste differentiation. So he and his colleagues recognized that when gynes emerge, that is future nest foundresses, they look a lot like as if they're about to enter diapause. And when workers emerge, they don't really look that way. And this probably has to do with differences in the nutrition that they received during development. But I'm bringing this up because I want to highlight this quote from the paper that reveals this assumption that is implicit in this hypothesis. And that is, these data suggest that gynes emerge in a state of reproductive diapause, by which caste differentiation could rely on regulatory circuits that in the ancestor controlled the conditional pre-pupal diapause pathway. So in other words, they're implying that the ancestral state was probably for something like on the figure I showed you on the last slide, where they're diapausing as pre-pupae. But somewhere along the axis of social evolution, there's been a shift to diapause in the adult stage. So this caught the attention of Dr. Santos and I, and we wanted to see if something like this might be happening in bees. So we went to the literature and found information about diapause for over 100 species of bees, and we found that absolutely none of the social bees diapause as larvae. In stark contrast to that, nearly all or most of the solitary bees diapause as larvae. We put this into an ancestral state reconstruction analysis, and sure enough, the ancestral state is predicted to be larval diapause, as indicated by these black nodes here. But in every case of an origin of eusociality in the bees, there's first been a loss of larval diapause. So this suggests that there's really something going on between the loss of larval diapause and sociality. And Jack De Silva followed this up a couple of years later, looking across all the Hymenoptera and found something very similar. There's been a loss of larval diapause prior to origins of eusociality in both wasps and bees. So what is this all about? How does a loss of larval diapause promote social evolution? Well, we are hypothesizing that the loss of larval diapause is promoting lifespan extension, allowing for eusociality to evolve. So remember that overlapping generations is one of the hallmarks of eusociality, because the queens are the mothers of her daughters who are the workers. And in solitary insects, or in solitary bees and wasps, most of them don't live long enough to meet their offspring as adults. And so before eusociality can evolve, there needs to first be an extension of lifespan so that mothers can overlap with daughters. And diapause can allow for the extension of lifespan by allowing insects to persist across unfavorable periods. Now, for example, if there were no diapause in this holometabolous insect, then once the unfavorable period comes, that's it. They're dead. That's the end of the life cycle. But if they have diapause, they can persist across that unfavorable period and thereby extend their total lifespan. Now, what's important about this is that diapausing as a larva enables the opportunity to get rid of any metabolic or respiratory damage that has occurred in the cells during that time, because larvae will undergo metamorphosis to pupae, and then the pupae once again will molt into adults. And with each of those stages of metamorphosis, there's an opportunity for cellular regeneration through cell division. In contrast, organisms that diapause as adults don't have that opportunity to renew their cells. So any sort of cellular damage that builds up during diapause, those individuals are stuck with that. All adult holometabolous insects are postmitotic. They will no longer generate new cells. And so this led Tadar and Yin to hypothesize that, well, then insects that diapause as adults should be under strong selection to reduce their senescence rate, to have a way to mitigate any sort of cellular damage that builds up during diapause, thereby leading to an extended adult lifespan, which is key for eusociality. Okay, so this led us to hypothesize that the loss of larval diapause promotes lifespan extension and thus facilitates social evolution. And we used a comparative genomics approach to test this with the prediction that we should see signatures of selection on molecular pathways that regulate lifespan and other life history traits. We collected 27 high quality bee genomes from across 6 families, and this represented at least 3 or more independent losses of larval diapause. We ran them through some standard comparative analyses, so looking for signatures of selection, comparing them to genes with a known role in oxidative stress and A gene mitigation, and then we looked at gene family expansion and contraction. So starting with those signatures of selection, we're able to identify close to 5,000 orthogroups. We looked at convergent rates of evolution, positive selection, and relaxed selection. And here are the results. So the number in each of these circles is telling you, or each of these partitions in this Venn diagram is telling you the number of orthogroups we found in this category. So for example, here are all of the orthogroups that showed a signature of relaxed selection. We overlapped them with the orthogroups that showed a negative rate or a decreasing rate of evolution convergent in the species that have lost larval diapause. And then we overlapped those with the orthogroups that showed signatures of positive selection in AGSx. And then, of course, here, the ones that showed convergent signatures of evolution, but with an increasing rate of evolution with the loss of larval diapause. And based on those, we could identify sets of genes that showed the best evidence for positive selection, the best evidence for purifying selection, and then possible genes that had a possible signature of positive selection, purifying selection, relaxed selection, or general intensified selection. And what we noticed is that the strongest signature here was relaxed selection. Most of these orthogroups were showing signatures of relaxed selection. And a decent chunk of them were in this insulin signaling TOR pathway. And that's significant because insulin signaling TOR, in its role as nutrient signaling pathways, they have direct effects on lifespan and growth. So you may be familiar with these caloric restriction experiments that lead to extended lifespan. And the reason for that is because it inhibits or reduces the activation of these nutrient signaling pathways that therefore can allow lifespan to be extended. Now, what's interesting, because these pathways are so well studied, we have a good understanding of their role in on lifespan. And we find that a lot of these genes that are under relaxed selection have a lifespan reducing component to or effect. And so that means that in the species that have lost larval diapause, these genes that reduce lifespan are under relaxed selection. Whereas in the genes or the species that have lost larval diapause, the genes that have a promoting effect on lifespan are under purifying selection. So we did a formal enrichment analysis. We looked at orthologs of genes that are involved in oxidative stress and orthologs of genes with a known function in aging. And we asked whether they're enriched in any of these categories. We didn't find any significant in enrichment for oxidative stress genes, although there's such a small number of them, it may be hard to detect. We did find a modest, though statistically significant, enrichment for aging genes among those genes that show an intensified response to selection. So selection's getting stronger on genes with a known role in aging. Okay, finally, we looked at gene family loss and gain using Kapheim. And we found one single set of genes, and that is a hormone that is conserved across insects and plays a role in ecdysone secretion, PTTH, and its receptor. So this PTTH and torso are known to influence prepupal diapause in a number of species and also metamorphosis through its role in stimulating ecdysone. Interestingly, these two genes are lost in every species for which larval diapause has been lost, except for one. And when we see that lost, it's concomitant with signatures of positive selection on this alternative tyrosine kinase pathway that also stimulates ecdysone secretion. We weren't entirely surprised to see PTTH and torso show up in this analysis because we had seen this paper from Costa et al. A couple years ago where they showed that PTTH and torso have been lost in the social bee genomes. We decided to look at this also, and it turns out that this result holds up. Here we're showing you the 27 bee genomes that we looked at. Again, the dark blue squares are the ones that have larval diapause. The dark purple squares tell you whether they have PTTH or not. So if it's purple, then they do have PTTH and torso. And then the dark gold squares are the solitary bees. And so you can see that these three traits are pretty covariant, right? So the species that have larval diapause also tend to be solitary and have PTTH and torso. But when you've lost larval diapause, you've also lost PTTH and torso, and this happens in all the lineages in which social behavior evolved. We wanted to investigate the relative roles of these two things in PTTH loss, so we used a phylogenetic least squares analysis, and we found the diapause stage explains a larger proportion of the variance in this pattern than does sociality. Remarkably, there's a recent paper showing, or preprint showing, that PTTH and torso have lifespan effects in Drosophila adults. So here I'm showing you some survival curves. These are figures from their paper. The red lines are the knockouts for PTTH, and over here the red line is a knockout for torso. And you can see in both cases, the flies that did not have PTTH or did not have torso lived significantly longer. They were also able to show that PTTH affects senescence rate in flies. So in the really old flies, the knockouts are able to climb up a vial in a shorter amount of time than the wild type. So again, our hypothesis is that the loss of larval diapause is promoting lifespan extension and therefore facilitating eusociality. And we found a couple lines of evidence to support this hypothesis. One, that there's been relaxed selection on the nutrient signaling pathways that affect lifespan. And there's been a loss of genes such as PTTH and torso that shorten lifespan. We also found some evidence for purifying selection on genes that extend lifespan. And with that, I just once again want to acknowledge my collaborator, Priscilla Santos, my excellent lab, funding from NSF, and support from the Utah Center for High Performance Computing. And if there's time, I'd be happy to take any questions. Thanks for your attention.
Thanks so much, Karen. I think we better move on to the next also social evolution talk, but please send your questions to the Q&A or contact Karen directly, and hopefully we'll have time after this to get a couple in live. So the next speaker is Dr. Sarah Kocher from Princeton University, and her topic is the molecular substrates of social evolution.
Hi, everyone. Thank you so much for tuning in, and thank you especially to the symposium organizers for giving me the opportunity to tell you about our work. So work in my research group centers on understanding the genetic and environmental factors that shape social behavior and its evolution. And to do this, we study variation in social behavior among the sweat bees, like the social species Laceglossum zephyrus that you see here. Social groups have arisen dozens of times across life on Earth, from cooperative interactions among members of microbial communities to the communal and social groups that are found in insects and many vertebrate species, including our own complex societies and culture. These behaviors are central to the survival and the success of these species, and they can help them to secure food, to secure mates, and to decrease their chances of predation. As a result, there's much interest in understanding the mechanisms that underlie the origins and the elaborations of this diverse set of behaviors. My research group is especially interested in what shapes the emergence and the breakdown of social behaviors. And as evolutionary biologists, we're keenly aware that studying organisms that vary naturally in these behaviors can provide us with a powerful lens to better understand what changes occur as social behaviors arise and evolve. And the social insects are, of course, a rich and diverse set of taxa that can vary extensively in social behaviors and take social living to the extreme, where we see within the bees, ants, and wasps eusociality, or the presence of a reproductive queen and a non-reproductive worker that interact to reproduce as a whole, has arisen about 7 different times within this group. So how does all this diversity in behavior arise? One of the major hypotheses that was put forth by Mary Jane West Eberhard is the ovarian ground plan hypothesis. And the basic idea is that the provisioning in the reproductive phases that are required for a solitary female to complete her life cycle have become parallelized as eusociality originated, and we now see the expression of these 2 different phases in 2 distinct castes, queens and workers. And since the ovarian ground plan has been proposed, there have been many subsequent elaborations and modifications to this basic idea, including the reproductive ground plan, which can be used to further explain division of labor among workers, as well as the diapause ground plan hypothesis, which posits that the genes and the gene networks associated with diapause have been co-opted during the evolution of eusociality, as well as developmental ground plan hypotheses that suggest that developmental plasticity and variation during development is an important component of eusocial evolution as well. And a more generalized version of these is the maternal heterochrony hypothesis, which argues simply that there were changes in the timing or the location of the gene networks that enabled the evolution of novel Cas, but it doesn't specify which types of genes. The most generalized hypothesis of all of these is the genetic toolkit hypothesis, which simply argues that there's a conserved set of genes that underlie the origins and the elaborations of eusociality. So given all of these amazing frameworks, we really wanted to know, if we look across different social and solitary species, can we identify shared molecular mechanisms associated with the evolution of social behavior within the social insects? And so to do this is work that was spearheaded by Burrell Jones and Ben Rubin, who were two former postdocs in the lab. And we collaborated with Olga Juchenko, who was a postdoc in the Aiden lab at Baylor. And we decided to focus our work on the sweat bees, which is the group of bees we study in my lab. And this is a unique group of socially variable bees, and they span the entire spectrum of social behaviors. So within the sweat bees, we have solitary species where each female occupies her own nest and is solely responsible for her young, as well as eusocial species that exhibit overlapping generations, cooperative brood care, and a non-reproducing worker caste. And there are even socially flexible species where females can produce either a solitary or a social nest, and this often depends on the environmental context that you can find them in. And the sweat bee life cycle is like that of many social insect species. You have single adult females that have overwintered that initiate their nests in early spring. And if they're solitary, they tend to provision a single brood that's a mix of males and females. And then the females mate, overwinter, and repeat the cycle again. And the difference between the solitary and the eusocial nests is that eusocial nests are again founded by a single foundress in the spring. But instead of provisioning a single brood that develops into reproductives, the foundress first provisions a worker brood that typically is female biased, and then those females help to provision a next round of brood where the eggs are still laid by the queen, and this includes the reproductive males and females. Then the females mate, overwinter, and repeat the cycle again. And among the social sweat bees, the colony sizes can vary quite a lot, but for most species, there's about 2 to 24 workers that are present inside a nest. There are species, however, like Laceglossum marginatum, where colonies can contain well over 400 workers. And so within this group, we have a lot of variation in social behavior that can be observed both within and between species. And there have also been multiple independent gains and losses of eusociality. So we know, thanks to work by Jason Gibbs and many other taxonomists, that there is a much larger phylogeny of sweat bees than the one that I've shown here. We can see through ancestral state reconstructions that there have been 2 independent origins of eusociality within this group. Once within the Algochlorinae about 25 million years ago, and once in the group that's associated with Halictus and Laceglossum, and this originated about 35 million years ago. In addition, there have been over a dozen independent losses within the group as well, which allows us to make comparisons between closely related social and solitary species within the same monophyletic family. So in order to identify the sets of genes that are linked to the evolution of eusociality in sweat bees, we first had to generate reference genomes for a set of about 20 focal species. And so we did this in collaboration with the Aiden lab. We used a combination of 10x linked reads and high-C sequencing so that we could create these chromosome level genome assemblies for the species. And just to give you an idea of what some of these data look like, we ended up with chromosome assemblies for each of the species that you see here. And on the right of your slide, you can see these bar plots that show you the assembly size and the estimated number of chromosomes from our data set. And the wedge plot that you see on your left shows you the chromosomes for our outgroup species. And the single colors inside each of the wedge correspond to a single color of a single chromosome in a different species. And so this should show you that there's conservation in synteny that's pretty well conserved across these different species. And if you're interested in accessing these resources, they're all online at beenome100.princeton.edu. So we've now been lucky enough to team up with the USDA Beenomes team, and we're proud to announce that we're up to 29 highly contiguous chromosome level genome assemblies for this group. So thank you so much to the Beenomes team for this. And we've also been able to get some assemblies from Darwin Tree of Life. So thank you to all of the people that have helped to make these resources broader for the group that we're studying. So we haven't yet had a chance to dive into the newest assemblies, so I'm going to focus on the results that we have from the first set of genomes that we assembled. And using these chromosome level assemblies, we limited our search to the one-to-one gene orthologs across each of these different species, so we could search for genes that show shared signatures of positive selection on the branches that represent these two independent gains of eusociality. Genes that exhibit convergent signatures of positive selection on each of these independent gains are going to show an increase in the number of amino acid substitutions on both branches where social behavior has arisen. And when we did this test, we found that there were 9 genes, which is about 4 times more than we would expect by chance, that show signatures of positive selection on both branches. And these include genes that are linked to glial cell ensheathing, like the gene shopper, and neurocognitive function, like the gene ND42. But most of the power in our SWEPB system comes from the replicated losses of social behavior. So we wanted to take advantage of this variation and these repeated losses within the SWEPBs to see if we could learn something about the mechanisms that are required for the maintenance of social traits within this group. And so the rationale is relatively simple. For example, if you imagine the ancestors of cave fish, as they moved into low light environments, these lineages began to rely less on their visual systems. And as a result, the selective constraints on the visual system became released and mutations began to accumulate that eventually led to the loss of function of the visual system in this group. Similarly, we were looking for genes that show complementary patterns of convergent relaxed selection within the SWEPBs, and these would have signatures of decreased intensity of selective pressures on all the branches where social behavior was subsequently lost. And when we did this test, we found 443 genes that show these convergent signatures of relaxed selection. And these genes are enriched for functions like chromosome condensation and vacuolar transport, which is usually related to the storage of lipids and other proteins in insects. So we were especially interested in genes that show convergent and complementary patterns of selection across these independent gains and losses. And the convergent positive selection on both branches implies that there are benefits associated with changes when eusociality is gained, while genes that show convergent relaxed selection suggest that there are costs associated with the maintenance of these traits when eusociality is lost. And therefore, genes that show both of these complementary signals would carry the classic signature of an evolutionary trade-off, suggesting that at those loci, beneficial changes tied to the origins of eusociality may convey a cost when eusociality is lost. And indeed, we found 4 genes that show the signature of convergent and complementary selection across all the gains and losses that we sampled. And that's almost a 9-fold enrichment compared to what we would expect by chance. So we lovingly call these genes in the lab the Fab4, and they include 3 genes that are well described, apolipoprotein, hexamerin 110, and PAT1, as well as 1 gene, OG11519, that doesn't have any clear orthologs outside of Hymenoptera and is of unknown function. And the fact that the Fab4 exists is very exciting because it suggests that comparative methods can help us to identify some of the least common denominators that are associated with social behavior. And the fact that we see these complementary patterns suggests that the mechanisms that are associated with the origin and elaboration of social behavior can also be identified by studying its loss. So it turns out that 2 members of the Fab4, apolipoprotein and hexamerin 110, are highly pleiotropic genes, but they play an important role in juvenile hormone binding and transport in insects. And juvenile hormone is an ancient hormone that's a key regulator of development and reproductive maturation in insects. And it turns out that changes in the levels of juvenile hormone that are circulating in the insect hemolymph have been associated with behavioral polymorphisms in all of the social insects that have been studied to date, but that the directions of these relationships can vary a lot across different species. And so although we understand that levels of juvenile hormone vary across many castes in many of these species, we know very little about the mechanisms that have been targeted by selection to change and modulate these hormone levels across these different behavioral forms. And our results are providing some new insights into how modifications to juvenile hormone binding and transport may have shaped the evolution of eusociality within the SWEPBs. Interestingly, studies from Drosophila have shown that one of these Fab4 binding proteins, apolipoprotein, crosses the blood-brain barrier with cargo intact, potentially delivering its cargo to the brain. And so we were curious about where apolipoprotein might be expressed in the SWEPB brain. And so we used single-cell RNA sequencing to characterize its expression patterns. And we found apolipoprotein and its binding partner apo-LTP are primarily expressed in a subset of glial cells that form and regulate the permeability of the blood-brain barrier in Drosophila. And in an orthogonal set of experiments that we were running in the lab, we were manipulating the early life social environments of bumblebees to look at the effects of social experience on the developing brain. And in those experiments, we compared gene expression profiles of bees that were either isolated in early life or that experienced limited or typical social interactions. And we discovered that isolated bees that have never encountered a social partner have reduced apolipoprotein expression compared to bees with typical or even limited social experiences. And this suggests that not only does apolipoprotein display signatures of selection across evolutionary timescales, but that we can also see changes in apolipoprotein expression linked to differences in the social environment during development. So this has led us to hypothesize that altering the availability and the uptake of juvenile hormone in the brain could be an important modulator of social behavior. And so we wanted to test this hypothesis. So we teamed up with some chemists that were down the hall from our lab, and we developed an isotope tracing assay where we could take a labeled form of juvenile hormone, topically apply it to the abdomens of the bees, and then sample the brains to see if we could detect movement of that hormone, that labeled hormone, into the brain following topical application to the abdomen. And when we did this, we found that indeed juvenile hormone is rapidly transported to the brain, and we can see that it peaks relatively quickly. So 4 hours post-treatment, we can see a peak in the amount of labeled juvenile hormone that we can detect. We also measured natural levels of juvenile hormone across solitary and social sweat bees, and we found that social queens have higher amounts of juvenile hormone in their brains compared to solitary reproductives, and that social workers had intermediate levels. And through work with collaborators, we've also found evidence that the regulation of juvenile hormone in the brain also modulates social behavior in ants. So in the study led by Lin-Yang Zhu and Carl Glastad in Shelley Berger's lab, they found that the blood-brain barrier acts as a hormonal gatekeeper. And through manipulation of a gene that can increase the permeability of the blood-brain barrier to juvenile hormone, you can measure subsequent increases of juvenile hormone in the brain, and this leads to higher rates of foraging in these individuals. This is amazing because it suggests that there's been convergent evolution on the brain hormone axis in both bees and ants, and this represents more than 180 million years of evolutionary divergence. And so together, our results suggest that altered hormonal uptake in the brain may be an important modulator of social behavior across both developmental and evolutionary time scales. And this, I think it's important to point out that now that genome sequencing is so accessible, it's become relatively common practice to be able to scan genomes and look for shared signatures of selection across eusociality. There's a number of great studies that hopefully you're hearing from other people about today that have also helped to give us insights into the shared set of molecular mechanisms that underlie social behavior and its evolution. And together, these generally support this idea that there's a genetic toolkit that underlies this variation. So to summarize the work that I told you about today, the unique variation in sweat bees can be used to provide insights into social evolution. And through our comparative genomic studies, we've been able to identify a core set of genes that are associated with the convergent gains and losses of social behavior within this group. And this work is pointing us to the hypothesis that altered hormonal uptake in the brain may be an important modulator of social interactions. And so with that, I would like to thank all of the members of the lab and all of the collaborators that have helped us to make these studies possible. Thank you to the Beenomes team and especially to the symposium organizers for inviting us to be here today. And thank you for listening. Thank you.
Okay, wow, those are two great talks on a hot topic for bees, of course, the social evolution. I think we're going to have to move on though to Kathleen Dogantzis' talk because we're running a bit behind. Please do put comments on the questions and we will get them to Sarah, and Karen is still on, so she will certainly address your comments. But our next speaker and final speaker will be Dr. Kathleen Dogantzis from York University, where she did a hugely productive PhD and is now doing postdoctoral work. And her topic is honeybee population genomics.
Hello, everyone, and thank you for attending my talk today. Today, I will be discussing how genetic variation has facilitated the adaptive radiation of 2 different honeybee species. So the genus Apis represents at least 12 different honeybee species. Most of the genus is native to Asia, with the exception of Apis mellifera, which has the largest distribution and encompasses Europe, Africa, and parts of West Asia. So contemporary species of Apis constitute 3 clades. In green, we have the dwarf honeybees, whose distribution is highlighted by the green coloring on the map. In blue, we have the giant honeybees, where some of the species distribution is highlighted in blue on the map. And then finally, there's cavity nesting bees, highlighted by both the red and purple coloration. Today's talk will focus on two cavity nesting bees, Apis mellifera and Apis cerana, the first of which I will be focusing on Apis mellifera. So divergence dating of the Apis genus using mitochondrial sequences estimates that the genus emerged about 10 to 30 million years ago. And Apis mellifera is hypothesized to have diverged from other cavity nesting bees about 7 million years ago. In this really nice study by G et al., applied a biogeographic reconstruction to the phylogeny of Apis in order to estimate the ancestral distribution of the species. So if we focus on the node that separates Apis mellifera from other cavity nesting bees, it is hypothesized that this divergence would have occurred somewhere in Southeast Asia, which is highlighted by this H on the map. After splitting from bees in Asia, it is hypothesized that Apis mellifera colonized its current distribution from Western Asia via 3 to 4 different expansion routes. So the first route is expected to have expanded into Africa, while there may have been a second route that colonized Northern Africa. It was expected that Europe was colonized via 2 separate expansions, the first into Eastern Europe, and then it's suspected that Western Europe was colonized by a second route via Northern expansion route. Since their colonization, the species has diversified into upwards of 30 different subspecies that can be delineated into at least 5 genetically distinct evolutionary lineages. So this includes the M lineage found in Western Europe as well as parts of Asia, the C lineage found in Eastern Europe, the O and the Y lineage found in West Asia, and the A lineage found throughout Africa. A recent population genomics study also identified a genetically distinct group in Egypt called the L lineage, which is composed of Apis mellifera lamarckii, as well as the U lineage, which is comprised of Apis mellifera unicolor from the island of Madagascar. Now, one of the most intriguing questions that arises from this pattern of colonization and evolution is how did lineages adapt to such a wide geographic distribution where environmental differences span between tropical and temperate environments? And this raises the question, how did genomic changes facilitate the adaptation of the species? And more specifically, can we identify mutations, genes, and potentially gene functions related to the adaptive radiation of Apis mellifera lineages? So this work is published in Science Advances, and here we discuss in a little more detail the origins of Apis mellifera, and what I'll be discussing now is the adaptation of the lineages themselves. So in order to answer this question, we worked with an extensive team of collaborators who were able to collect honeybee tissue and DNA from across the native range of Apis mellifera. Our final data set was composed of 251 individual genomes, 160 of which were newly sequenced, with the remaining samples coming from the studies we've listed down here. So here we extracted DNA from our 251 individuals and sent them from genome sequencing, where we identified almost 12 million mutations across the 251 samples. So the first objective of this study was to identify if there was any admixture among samples and to classify individuals into their respective lineages. So we used several population structure analyses to do this, including admixture and clustering using a PCA. We found that samples clustered into 7 genetically distinct groups. The majority of samples clustered into already recognized genetic lineages. So the A lineage of Africa, the C and the M lineage found in Europe, and the O and the Y lineages found in West Asia. However, as I mentioned in the intro, we also discovered 2 independently clustering lineages. So the L lineage, which is composed of Apis mellifera lamarckii from Egypt, and the U lineage, which is comprised of Apis mellifera unicolor from the island of Madagascar. And these 7 groupings were the foundation of this study. So in order to identify mutations in genes that may be associated with the adaptation of Apis mellifera, we wanted to identify patterns of positive selection, as positive selection is considered to be the primary mechanism for adaptation. So in order to identify positive selection, we use outlier measures of FST, which compares the allele frequency between groups or different populations. So the idea here is that when there's a new mutation that arises in a population, if that mutation offers a fitness benefit to that population, over time that allele or that mutation should increase in frequency within the population. Such that if we compare two populations, so one with the novel mutation that offers a fitness effect versus a population that does not have that novel mutation, we should find that areas under selection should be highly differentiated when we compare them to regions not under selection. So here we calculated pairwise measures of FST between each lineage and examined the genomic distribution of FST measures in order to identify loci within the upper 95th percentile of that distribution. So for example, if this is our focal population, we calculate FST at each variable site between each of the pairwise comparisons. We graph those measures of FST and then target the most highly differentiated sites among those comparisons. And more specifically, we're looking for variants that are consistently within the upper 95th percentile for all pairwise comparisons, and we deem those to be lineage-specific outliers. So these are variants that are highly present or in high abundance in the focal population, but then present at low frequency within the rest of the lineages. So here we have the breakdown of the number of outlier markers associated with each of our lineages. You'll see that the M lineage was divided into two groups, so samples from Europe and samples from Asia. And you'll also notice that the A lineage had very few outlier markers, so we actually excluded it from further analyses in the study. Okay, so now that we have an idea of how many unique outliers are associated with each of these lineages, we wanted to know how these mutations are actually distributed across the genome and if they're associated with any particular genes. And more specifically, we're interested in if the mutations are enriched or found in abundance in functionally relevant portions of the genome. So for example, mutations in coding regions that potentially affect the protein structure and function of genes, while mutations in regulatory regions may be influencing gene expression levels. So when we focus on unique lineage mutations associated with positive selection among the promoter region, which we've defined as 1,000 base pairs upstream of the gene's start codon, we find that on average there's about a 20% enrichment of mutations among this regions. So the direction of the arrow indicates if the enrichment is increased or depreciated, and the coloring of the arrow indicates that that enrichment was significant relative to the gray arrow, which indicates that the enrichment was insignificant or non-significant. Among exons, we find that SNPs associated with positive selection are enriched on average 26%. And we find an increase in enrichment among all lineages with the exception of two groups. And finally, when we look at the distribution of SNPs associated with selection among introns, we find that they are deficient of outlier SNPs, which is significant among 4 of the lineages, and this decreases about 3% overall. So overall, we find that SNPs under selection are clustered among functionally relevant regions, suggesting they may indeed have an influence on the adaptation of these bees. We were also interested in functions associated with loci under selection, and a gene ontology analysis highlights broad categories related to morphogenesis, development, and behavior. And this pattern of selection is consistent with the extensive morphological and behavioral adaptations that have developed among the species, especially because their distribution is so wide and they encompass such different ecological niches. However, what's even more interesting about SNPs under selection is that although each of these SNPs is distinct to a particular lineage, we find that their distribution was concentrated among a relatively small set of genomic hotspots. So more specifically, there was about 145 genes that contained at least one outlier SNP that was common across all of the honeybee lineages we looked at. Some of these genes have been found to be associated with things like response to varroa, colony defense, and the production of royal jelly and honey. But the result of genomic hotspots or repeated selection among genes has also been noted among Apis cerana. So this really nice study by G et al. Measured signatures of selection among distinct Apis cerana lineages in China. And though they note that selection among genes was skewed towards lineage-specific genes, there are a few genes that were repeatedly under selection in more than one lineage. And if we focus on this gene here, they found that the leukocaine receptor gene, which is hypothesized to be related to foraging division of labor, was under selection in each of the lineages they looked at. So this emerging pattern of gene reuse among apis to facilitate rapid adaptation and rapid radiation is really interesting, and I'm excited to see if these patterns are observed in other apis species in the future. So finally, we were interested in whether the queen or the worker phenotype has a disproportionate role on the adaptation of lineages. And we did this by comparing genes under selection to those differentially expressed between queen and worker castes. So we used two previously published datasets to do this. So the first focused on differentially expressed genes developing in queen and worker larvae. So in total, we found just under 1,200 genes that were upregulated in the queen caste relative to the worker caste, and approximately 2,000 genes that were upregulated in the worker caste relative to the queen caste during the development stage. We also worked with a previously published data set that looked at the differential expression of proteins between queens and workers. And in total, we had 77 genes expressed among queens relative to 90 genes expressed in workers relative to queens. And what we discovered was that genes associated with positive selection among lineages were significantly enriched in the worker caste relative to the queen caste. And the worker caste is highlighted in this dark blue color. On average, there was a 95% increase of genes under selection relative to expected values among developing larvae, while we found a 180% increase of genes under selection relative to expected values among adults. So relative to the queen caste, which is highlighted in this light blue color, we actually observed an underrepresentation of genes under selection, equating to a decrease of 42% among developing larvae and a 33% decrease among adults. So in summary, we find that outlier SNPs are enriched among protein coding and promoter regions, which was significant among most lineages, suggesting that protein coding changes in gene regulation are hotspots for selection. We also discovered that the functions of genes under selection were associated with colony-level traits such as aggression and pathogen response, suggesting that many of the same phenotypes are involved with adaptation among lineages. And then finally, genes under selection are enriched among differentially expressed genes and proteins among the worker caste, suggesting that the worker phenotype has a disproportionate role in adaptation among Apis mellifera lineages. While it is evident that genetic diversity among Apis mellifera lineages facilitated the adaptive radiation of the species to their very wide distribution, these changes are suspected to have occurred potentially over hundreds of thousands of years. So what I would like to transition to now is a story about Apis cerana, where the species has quickly adapted to a new area over a very short 10-year period of time. And this work was recently published in collaboration with the University of Sydney. And shout out to Ross Glogg, who I worked really closely with on this project. So Apis cerana is endemic to most of Asia and is comprised of about 8 different subspecies. In the 1970s, colonies from the subspecies in Java were intentionally introduced to New Guinea. And then by the early 1990s, these colonies had spread across New Guinea into the surrounding islands. And in 2003, Apis cerana was detected on the Solomon Islands. In 2007, a reproductive swarm was discovered in Cairns, Australia. And as part of a biosecurity response, eradication of the colonies was attempted. And during this time, population sampling was conducted from their point of introduction to about 2018. So during this time, the species expanded to about 10 to 50,000 colonies across a range of 10,000 kilometers squared. But because of these sampling efforts, we have this really nice timeline showing the first 10 years of the incursion. And we're able to sequence these individuals in order to study and document whole genome temporal changes, including changes in allele frequency and the genomic diversity. So in addition to these samples in Australia, we were also able to work with collaborators to collect samples from the progenitor populations. So this includes the source population located in Java and the introduced population of New Guinea. So one of the most striking things to note about this data set is that the sequential introductions from Java, then to New Guinea, and then to Australia has produced a series of bottlenecks that has reduced the genetic diversity with each introduction. So here we show measures of nucleotide diversity and heterozygosity of both the Australian populations and the progenitors. And here we note that the population in Java shows the highest level of diversity across both of these measures. And then with each introduction, we see this stepwise decline in overall genomic diversity. But given the substantial success of the invasive Apis cerana population in Australia, it raises this really interesting question of how, despite experiencing a significant bottleneck, how was the incursion so successful? And this sort of ties back to the same question we posed in the previous study of how did genomic changes facilitate the adaptation of the species? And more specifically, can we identify mutations and particular genes that may be related to the successful adaptive radiation of Apis cerana in Australia? First, we wanted to identify patterns of positive selection. As I mentioned earlier, positive selection is considered the primary mechanism for adaptation. And to do this, we use measures of selection that focus on changes in allele frequency. The first set of measures we looked at are often associated with selective sweeps, which is a process that reduces variation around the site of selection. So for instance, we can expect diversity to decrease at sites under selection. We can expect measures of Tegemus D to decrease at sites under selection. And we expect linkage disequilibrium between mutations to increase at sites under selection. We are also interested in targeting regions under new directional selection or selection that is divergent from which may have been present in the New Guinea population. So thus, we were also looking to target regions with high measures of differentiation between the Australian population and the progenitor population. So using this method, we identified 876 regions with signatures consistent with the 4 measures of positive selection. So here we note that regions under selection, which are highlighted in orange, show an increase in differentiation relative to the genomic background, which is highlighted in this blue dashed line. Our outlier regions also had lower measures of Tegemus D relative to the genomic background. They had a reduction in diversity relative to the genomic background and also showed elevated levels of linkage disequilibrium relative to the genomic background. And overall, these patterns suggest that we're on the right track for identifying potential regions under positive selection. So finally, we wanted to further refine regions under positive selection by trying to identify particular mutations that may be the drivers of selection at these particular sites. So we target SNPs using two different methods. So the first approach uses measures of population differentiation to detect outlier SNPs. So as I mentioned earlier, beneficial mutations are expected to increase in frequency among the selected population, such that levels of differentiation should be high at selected sites relative to unselected sites. So if we compare a New Guinea population and Australia population, sites that are under directional selection in Australia should be highly differentiated from those in New Guinea. The second method here used the temporal changes in allele frequency, where it's hypothesized that low standard directional selection will show positive correlations between the changes in allele frequency and the time series data we have over this 10-year period. And then relative to neutral loci, which are presumably affected by drift alone and no selective forces, should have values closer to 0. So we're looking for SNPs that show a nice correlation between our allele frequency and time. So in total, we identified 480 SNPs that fit our selection criteria and were associated with a putative functional effect. So for example, they were located within a regulatory region or within a protein coding region. On this graph here, we show changes in allele frequency for these highly differentiated SNPs in red. And then in blue, we show the allele frequency changes for SNPs that follow this nice correlation over time. The green portion of the graph shows the SNPs that overlap between both methods. But the major thing I want you to take away is that SNPs under selection were already present in the progenitor population, suggesting that selection is occurring on standing genetic variation as opposed to novel mutations, and that despite a bottleneck, the current population has enough genetic diversity for adaptation to occur. Right, so finally, we were interested in the functions of genes under selection and find three main categories. So the first is that we find genes associated with selection are associated with the caste development of queens and workers. And more specifically, these genes are associated with differential expression during the developmental phase. We also find several genes related to reproduction, including a gene found to be expressed in the spermatheca of mated queens relative to virgin queens. And then finally, we find a gene related to worker behavior. And if we take a closer look, we note that that gene is the leukokinnin receptor gene, which is hypothesized to regulate foraging division of labor between pollen and nectar. And as a reminder, this is the same gene that was found to be under repeated selection among Apis cerana lineages in China. And it's really interesting to see this gene come up over and over again and to repeatedly see this trend of gene reuse. So this gene may play a prominent role in enabling rapid adaptation to diverse habitats, especially maybe trying to use different floral resources. And thus it may have enabled Apis cerana in Australia to readily adapt to its new environment, much like the patterns we're seeing of Apis cerana in other geographic regions. So in summary, we find that genetic diversity among the invasive population is greatly reduced, showcasing that with each sequential introduction, a bottleneck has contributed to the reduction in overall genetic diversity. Selection in the Australian population seems to be occurring on standing genetic variation relative to novel mutations, and this genetic variation is suspected to have come from the progenitor populations. And this suggests that despite having a genetic bottleneck and seeing this reduction in diversity, there is sufficient variation for adaptation to occur within this particular population. Finally, genes under selection seem to be related to development and worker behavior, and we detect selection on genes known to be associated with gene reuse among Apis cerana. And again, in the future, it'd be really interesting to see if this idea of gene reuse seems to be prominent among other Apis species. And with that, I would just like to thank everyone who's been involved with these projects. None of this work would have been possible without the contributions of these individuals. Again, I just want to have a big shout out to Ross Glog from the University of Sydney, who I worked really closely with on the APIS cerana project. And of course, all the funders and institutions involved with these projects.
All right, thank you so much, Katie. That was fantastic. There is a little time, I think, for questions, and I think also our speakers from prior groupings are available if you have ones going back. I just had a quick, maybe more, thinking as an applied honeybee person, the leukocynin receptor and its neuropeptide, is that something that you could measure at the expression level that might be tied to hygienic behavior? Or is it just something that's historically pushed by different behavioral trends in bees?
So I think, I mean, we probably could measure it. I think that paper by G, they did like a knockdown to actually like test the effects of the leukocynin receptor. Off the top of my head, I don't remember the details of it, but it has been looked at, yeah.
Yeah, but there's something but your work suggests something about its own sequence or maybe binding capacity to the neuropeptide or something is what's.
Yeah, so either, I think like for that gene in particular, it may have been like associated, like SNPs associated with regulatory regions more so than like protein coding changes. So it probably does tie into some differences in expression levels. We didn't like empirically test that, but that would be like the working hypothesis, I guess.
Cool. Any other questions for the speakers? Katie, I can bug you later with more honeybee specific ones, but one other thing, I know you looked at the subspecies or races, as it were, of honeybees. Have you also looked at, very clearly artificially selected lines of bees in commercial beekeeping versus more feral, more wild ones? And do they show a similar, whether it's immune traits or these behavioral traits, do they show different positive selection versus things that weren't being pushed by humans?
That's a good question. I haven't compared like commercial to feral. I have done a little bit of work looking at admixture in like commercial Canadian colonies and to see like how admixture may affect like traits overall. And we do find in regions where there's like preferential admixture for one of the lineages that make up the populations, we do find genes related to like olfaction and varroa response. But beyond that, I haven't taken a look. Yeah.
Cool. All right, and again, anyone who's still on, remember the talks. Anna put up, I believe, the link to the talks, which will be on YouTube, as long as YouTube lasts. So please check those out in a couple weeks. Thanks to Pia Olofsson, who's been coordinating all the logistics of that. Any other final, Michael, any final words? Oh, something popped up. There was a quick question about ants and their chemical signals for companions. Sort of I think the trail marking traits of ants. And I don't know, maybe Michael as another ant/bee person might know the answer to that.
I mean, I think it's wondering if bees are like ants and using a trail pheromone. And I think the answer is very much no, because one, most bees are solitary, so they're not, communicating with other members of a colony. But then for the social lineages, I mean, I actually don't know this question. Maybe Sarah, if she's here, or Karen would know better. But I think like with honeybees, right, it's less about, detecting pheromones and more about visual cues. Since they're not walking, they're flying. But I think we do know at least that there's some chemical cues for finding nests, I guess. Even solitary bees will use pheromones to help them, locate their, their stem nests. And I imagine other social bees use, similar strategies, but I don't know the details.
Great. Yeah, thanks for the question. I think Dr. Raymond is in Malaysia. It looks like another one for Katie just popped up.
Okay, so the question is, I'm wondering if your sample size could affect your outlier detection, especially when you split between workers and queens. So this is a good point. So it definitely did for our A lineage, so being able to detect outliers among that group. So we did the analysis because we did have so many more individuals for this lineage. We did pare down the analysis to just 10 samples to see if that would affect the measures. It did not, and we think a lot of that has to do with the effective population size of the lineage itself affecting the measures of FST we used. And then I'm not quite sure, we mean split between queens, workers and queens, maybe if you're referring to like the expression analysis. So that those data points were generated from a previous study, and I think there was about 10 or so individuals per group. So I don't think it would have had too much an effect on the actual expression results. Hopefully that answers the question though.
Great, thank you, Katie. All right, and thanks to everyone for hanging in there, and to Brad and Pia and Glenn for running the show, as always. If you're interested, there's one final session in this series, which is by our colleagues at the NCBI at NIH. We'll be talking about comparative genomics, and they're great resources that can be used by us insect people. So that's May 21st.
Yeah. And last but not least, thanks to Jay for running the show as well, kind of being the moderator and mediator all the way through. Appreciate all your work, and as well as Michael for helping organize this as well.
Yeah, great talks, everyone. Thanks.
Thank you, everybody.
All right, bye, everybody.
Take care. Yeah, bye.